Carbohydrates chemistry. In this chapter, we discuss nomenclature, structure, and function of monosaccharides, disaccharides, and the major homoglycans, starch, glycogen, cellulose, and chitin. We then consider proteoglycans, peptidoglycans, and glycoproteins, all of which contain heteroglycan chains. Carbohydrates, also called saccharides, are, on the basis of mass, the most abundant class of biological molecules on Earth. Although all organisms can synthesize carbohydrate, much of it is produced by photosynthetic organisms, including bacteria, algae, and plants. The name carbohydrate, hydrate of carbon, refers to their empirical formula, CH2O, N, where N is 3 or greater. Carbohydrates can be described by the number of monomeric units they contain. Monosaccharides are the smallest units of carbohydrate structure. Oligosaccharides are polymers of 2 to about 20 monosaccharide residues. The most common oligosaccharides are disaccharides, which consist of 2 linked monosaccharide residues. Polysaccharides are polymers that contain many, usually more than 20 monosaccharide residues. Monosaccharides. Monosaccharides are water-soluble, white, crystalline solids that have a sweet taste. Examples include glucose and fructose. Chemically, monosaccharides are polyhydroxyaldehydes, or aldoses, or polyhydroxyketones, or ketoses. They are classified by their type of carbonyl group and their number of carbon atoms. The smallest monosaccharides are trioses, or 3-carbon sugars, 1 or 2 carbon compounds having the general formula, CH2O, and do not have properties typical of carbohydrates, such as sweet taste and the ability to crystallize. The aldehydic triose, or aldotriose, is glyceraldehyde. Glyceraldehyde is chiral because its central carbon, C2, has 4 different groups attached to it. The ketonic triose, or ketotriose, is dihydroxyacetone. It is achiral because it has no asymmetric carbon atom. All other monosaccharides, longer chain versions of these 2 sugars, are chiral. The stereoisomers D and L-glyceraldehyde, the form of glyceraldehyde that caused rotation to the right, dextrorotatory, was designated D, and the form that caused rotation to the left, levorotatory, was designated L. Longer aldoses and ketoses can be regarded as extensions of glyceraldehyde and dihydroxyacetone, respectively, with chiral HC, OH groups inserted between the carbonyl carbon and the primary alcohol group. The complete list of the names and structures of the tetroses, 4-carbon aldoses, pentoses, 5-carbon aldoses, and hexoses, 6-carbon aldoses, related to diglyceraldehyde. Many of these monosaccharides are not synthesized by most organisms. The carbon atoms are numbered from the carbon of the aldehyde group that is assigned the number 1. By convention, sugars are said to have the D configuration when the configuration of the chiral carbon with the highest number, the chiral carbon most distant from the carbonyl carbon, is the same as that of C2 of diglyceraldehyde. The arrangement of asymmetric carbon atoms is unique for each monosaccharide, giving each its distinctive properties. Except for glyceraldehyde, which was used as the standard, there is no predictable association between the absolute configuration of a sugar and whether it is dextrorotatory or levorotatory. It is mostly the D enantiomers that are synthesized in living cells, just as the L enantiomers of amino acids are more common. The L enantiomers of these 15 aldoses are not shown. Recall that pairs of enantiomers are mirror images. In other words, the configuration at each chiral carbon is opposite. For example, the hydroxyl groups bound to carbon atoms 2, 3, 4, and 5 of D-glucose point right, and right, respectively in the Fischer projection. Those of L-glucose point left, and left. The 3-carbon aldose, glyceraldehyde, has only a single chiral atom, C2, and therefore only 2 stereoisomers. There are 4 stereoisomers for aldotetroses, D-N-L-erythrose and D-N-L-threose, because erythrose and threose each possess 2 chiral carbon atoms. In general, there are 2 N possible stereoisomers for a compound with N chiral carbons. Aldohexoses, which possess 4 chiral carbons, have a total of 24 or 16 stereoisomers, the 8D aldohexoses and their L enantiomers. Sugar molecules that differ in configuration at only one of several chiral centers are called epimers. For example, D-mannose and D-galactose are epimers of D-glucose, at C2 and C4, respectively. Although they are not epimers of each other, longer-chain ketoses are related to dihydroxyacetone, in the same way that longer-chain aldoses are related to glyceraldehyde. Note that a ketose has 1 fewer chiral carbon atom than the aldose of the same empirical formula. For example, there are only 2 stereoisomers for the 1-ketotetrose, D-N-L-erythrulose, and 4 stereoisomers for ketopentoses, D-N-L-xylulose and D-N-L-ribulose. Ketotetrose and ketopentoses are named by inserting -ol in the name of the corresponding aldose. For example, the ketose xylulose corresponds to the aldose xylose. This nomenclature does not apply to the ketohexoses, tagatose, sorbose, cyclose, and fructose, because they have traditional, trivial names. Cyclization of aldoses and ketoses. The optical behavior of some monosaccharides suggests they have one more chiral carbon atom, diglucose, for example, exists in two forms that contain 5, not 4, asymmetric carbons. The source of this additional asymmetry is an intramolecular cyclization reaction that produces a new chiral center at the carbon atom of the carbonyl group. This cyclization resembles the reaction of an alcohol with an aldehyde to form a hemiacetal, or with acetone to form a hemiketal. The carbonyl carbon of an aldose containing at least 5 carbon atoms, or of a ketose containing at least 6 carbon atoms, can react with an intramolecular hydroxyl group to form a cyclic hemiacetal or cyclic hemiketal, respectively. The oxygen atom from the reacting hydroxyl group becomes a member of the 5 or 6-membered ring structures because it resembles the 6-membered heterocyclic compound pyrene. The 6-membered ring of a monosaccharide is called a pyranose. Similarly, because the 5-membered ring of a monosaccharide resembles furan, it is called a furanose. Note that, unlike pyrene and furan, the rings of carbohydrates do not contain double bonds. The most oxidized carbon of a cyclized monosaccharide, the one attached to 2 oxygen atoms, is referred to as the anomeric carbon. In ring structures, the anomeric carbon is chiral. Thus, the cyclized aldose or ketose can adopt either of 2 configurations, designated alpha or beta, as illustrated for D-glucose. The alpha and beta isomers are called anomers. In solution, aldoses and ketoses that form ring structures equilibrate among their various cyclic and open-chain forms. At 31 degrees Celsius, for example, D-glucose exists in an equilibrium mixture of approximately 64% beta-D-glucopyranose and 36% alpha-D-glucopyranose, with very small amounts of the furanose, figure 8. 9 in open-chain forms. Similarly, D-ribose exists as a mixture of approximately 58.5% beta-D-ribopyranose, 21.5% alpha-D-ribopyranose, 13.5% beta-D-ribofuranose, and 6.5% alpha-D-ribofuranose, with a tiny fraction in the open-chain form. The relative abundance of the various forms of monosaccharides at equilibrium reflects the relative stabilities of each form. Although unsubstituted D-ribose is most stable as the beta-pyranose, its structure in nucleotides is the beta-furanose form. Conformations of monosaccharides. Haworth projections are commonly used in biochemistry because they accurately depict the configuration of the atoms and groups at each carbon atom of the sugar's backbone. Cyclic monosaccharides can exist in a variety of conformations, 3-dimensional shapes having the same configuration. Furanose rings adopt envelope conformation CN, which one of the 5 ring atoms, either C2 or C3, is out of plane and the remaining 4 are approximately coplanar. Furanoses can also form twist conformations where 2 of the 5 ring atoms are out of plane, one on either side of the plane formed by the other 3 atoms. Pyranose rings tend to assume one of 2 conformations, the chair conformation or the boat conformation. There are 2 distinct chair conformers and 6 distinct boat conformers for each pyranose. The chair conformations minimize steric repulsion among the ring substituents and are generally more stable than boat conformations. The H, OH, and CH2OH substituents of a pyranose ring in the chair conformation may occupy 2 different positions. In the axial position, the substituent is above or below the plane of the ring, while in the equatorial position, the substituent lies in the plane of the ring. In pyranoses, 5 substituents are axial and 5 are equatorial. Whether a group is axial or equatorial depends on which carbon atom, C1 or C4, extends above the plane of the ring when the ring is in the chair conformation. The more stable conformation is the one in which the bulkiest ring substituents are equatorial. In fact, this conformation of beta-D-glucose has the least steric strain of any aldohexose. Pyranose rings are occasionally forced to adopt slightly different conformations, such as the unstable half-chair adopted by a polysaccharide residue in the active site of lysozyme.